like to welcome you all to the May Early Onset Parent Roundtable. We have a very special team of guests this evening with us, led by our very own Heloise Hoffmann. A lot of people in the FSHD community are familiar with Heloise because she is such a superstar in our community. She is a sophomore at Stanford, where she is leading the international genetically engineered machine team, and they're working on hopefully a cure to FSHD. I'll let her talk more about that because again, it's going to be way over my head. But we're so happy to have them with us. Like I was just saying, this meeting is recorded and you can view it later on the FSHD Society webpage. And I will turn it over to Heloise. Heloise, thank you so much for agreeing to be here today. I know you're super busy with the end of the school year and all that kind of stuff. And I know that you have a very tight schedule. So we very much appreciate you being here with us.
It's such a joy to be here. Thank you so much for having us. I know the team is really excited to be here, and we're very honored to get the chance to hop on today. And so I have some slides that I can share. I'm not sure if I have access. Let me.
Heloise, do you want to introduce your team real quick before you get started so that people are familiar with who they are?
Absolutely. And I'll have the chance to introduce ourselves to once I get the slides up. But just a quick overview is that we're the Stanford iGEM team, and we're working on a treatment for FSHD right now. We just started working in January, so we're really excited to ramp up the research over the next couple weeks and months, and get some real progress done. So I'm going to start sharing my screen. Perfect. Thank you, Alice.
Of course.
All right. So as I mentioned, we are the Stanford iGEM team, and we've been working for the past couple months on trying to develop a new therapeutic for FSHD, which is really exciting. And the wonderful Muffer who's on the call today has designed a lot of these cute graphics, so shout out to her. But we can go to the next slide. So here's a team photo that we took at a recent symposium that we attended, where we just presented some of our preliminary project ideas. And now we've got a firmer idea of what we're doing. So a very exciting time. And we're all undergrads at Stanford here, working through a global program called iGEM that's focused on enabling accessible undergraduate research around the world to help empower students to pursue research topics that they're really excited about. And we'll get the opportunity in the fall to actually travel to Paris to present this research, which will be such a wonderful opportunity to raise awareness for FSHD as well on the national level. So now we can kind of introduce ourselves individually. If we can go to the next slide. So my name is Heloise, and some of you may know me. I've been pretty active with the FSHD Society over the past years. I started my advocacy journey around 5 years ago, which seems like a completely different time now. I was diagnosed with FSHD when I was 13, which has really been the driving force for my advocacy. And symptoms really started showing up around 10. So I feel really strongly about empowering the early onset community as well, because, just thinking about when I was diagnosed at 13, there's still no treatment today, but one day there will be. And to think that there could have been a treatment on shelves at that time, but I would be denied taking it because of my age, even though, the early onset population is the population that needs it most, that's just devastating. So I'm very committed to working with this group to ensure that we have an accessible treatment available to all in the next short years. So that's really my driving force. And I've been really active with the FSHD Society, leading the Gen Z of FSHD group, and also founding the Southwest Florida chapter of the FSHD Society. And now I've really focused a lot of attention on research because I discovered a love for biochemistry and molecular biology and realized that I could hopefully really use that to accelerate a treatment. So that's kind of why I'm here. And that's what really excites me about this project and this opportunity, is that it's a chance to accelerate a cure, but also give hope to the FSHD community, because I know that for me, the hope for treatment is really what keeps me going every day. And so to be able to be part of that in such a hands-on way is really exciting. So yeah, I'll hand it to the next member of the team.
Yeah, awesome. Thank you so much, Eloise. Yeah, so my name is Alice. I'm also considering majoring in bioengineering, and I'm a freshman at Stanford, and I'm from the Bay Area, so pretty close by. And then I love dancing and making jewelry. Those are my fun fact. And then for me, part of the reason why I'm so excited about this project is that we can build off of existing tools in bioengineering and biology and apply them to help the lives of those with FSHD. And just, it's been so inspiring, like learning about all the pathology behind the disease, but also how much potential there is for innovation. And so I'm really excited to push that through with this team. And I can't wait to work full time over this summer.
I'm Emmanuel. I'm from Kansas. I'm also considering majoring in bioengineering. When I first heard Eloise's idea in January for what we should do as a project, I thought that was a fantastic way that we should spend our time. And it seems like just like an amazing way to make a difference. So, yeah.
Hi, everyone. My name is Vanessa. I'm another one of the team members for iGEM. I'm a sophomore at Stanford from Southern California. Fun fact is I know the entire script of Ratatouille. And I guess like something that like just really excited me about this project was like just tackling this global challenge, FSHD, with like novel synthetic biology, like biosynthetic pathways, but also having like this like interdisciplinary like nature where we can incorporate like computer science and like computational like modeling and more many other focuses to this project beyond just like biosynthetic pathways. So yeah.
Hello, everyone. Thank you so much for having us here. My name is Maffer. I'm a freshman at Stanford studying electrical engineering and earth systems. I'm originally from Lima, Peru. And my fun fact is that I have not watched any High School Musical movie yet, but we probably will during the summer, hopefully. And what excites me the most about iGEM and the project in general is the endless possibilities to combine science and technology to create a solution that actually matters and that creates impact, because that's at the end of the day the part of science, changing lives. So that's something very exciting to me, and I cannot wait to work in weekend this summer.
And Michael is actually able to join us, so if you want to introduce yourself.
Yes. Hello, my name is Michael. I'm also thinking of studying bioengineering at Stanford. I guess a fun fact would be I'm really into cosplaying. And the reason why, like, I'm very excited to work on this project because after reading all the papers and getting updated on the current research, it feels like breakthrough is just like around the turn and around the corner, and I hope we can make that turn. And yeah, just bring this therapeutic would be very exciting. I'm excited to work with you guys over the summer.
Yes. And just want to highlight two of our additional members, Ayushi and Catherine. Unfortunately, they had midterms and classes because we're still in full swing for the quarter, but they are also such wonderful members of our team, so wanted to give them a shout out as well. If we can move to the next slide. All right, so now that you've gotten a chance to know our team, we want to give you a brief overview of our actual research project, which is the really exciting part. So we just started working a few months ago, as I mentioned earlier, and so we're just kind of in the beginning phases of getting the ball rolling in lab, but we do have our idea fully fleshed out. So we're going to give you just a brief overview of our project and hopefully get you excited about some of this new research that we're doing here at Stanford on FSHD. But before we dive into the actual project, I wanted to give a refresher on FSHD biology. So at this point, you might have all watched some of the FSHD Society videos with the biology behind FSHD and the molecular mechanism behind DUX4 expression, with the classic analogy with the cookbook. I know that's how I learned the biology behind FSHD. So I won't I won't beat a dead horse with that. But I want to start at the point where DUX4 gets expressed. So at this point, you are probably familiar with the fact that DUX4 is the toxic protein that's at the root of FSHD pathology. So the important thing to note is that DUX4 is actually a transcription factor, and it should be silenced. In other words, it should not be expressed in healthy people, but because of two potential genetic mutations in FSHD, DUX4 is expressed, and that's what causes muscle death. And so a little bit more about what a transcription factor is, because that's really what our approach centers around, is basically DNA is organized in these long strands, and there are many gene blocks, one after the other. And just before the coding region of a gene, there's actually something called a promoter, which is drawn in purple there, where that's actually a binding site for these proteins called transcription factors, which DUX4 is one of those. And there are two different flavors of transcription factors. You have repressors and activators, but we'll focus more so on the activators because DUX4 typically acts as a transcriptional activator. So essentially, in most cases, you have these genes in blue on this diagram that I quickly drew up, and they're located just behind this promoter region. And if a transcription factor doesn't bind to that promoter region, that gene won't be expressed and will be generally silent. However, when a transcription factor does bind to that region, that gene will be expressed and turned into protein, because that transcription factor actually signals to the cell to bring a bunch of ingredients, to kind of follow this cookbook analogy, to that DNA and read that recipe. And so that's where DUX4 comes in, and that's why I drew that little duck there, because DUX4 actually acts as a transcription factor to start the transcription of hundreds of downstream genes that should really be silenced. Next slide. So just to kind of recap all of that and fit it into the context of FSHD, DUX4 should be silenced in healthy individuals, but unfortunately, in the case of FSHD, it gets expressed due to a genetic mutation. And then it itself actually goes on to activate hundreds of genes that should also be silenced, which then lead to a lot of inflammation and accumulation of these molecules called cytokines, and basically signals to cells, to muscle cells, to start apoptosis, which is basically a cell suicide. And essentially, all of this whole pathway leads to muscle degeneration. As you can see in this diagram, on the left, you have healthy muscle, and then on the right, this is a biopsy of someone with FSHD, and you can see all of the gaps between those muscle fibers, and that muscle really gets broken down. And so that's kind of how FSHD happens. And so that's what we're trying to address. So next slide. Going back to this idea of transcription factors, as I mentioned, DUX4 is a transcription factor itself in the homeobox family. So this is just a interpretation of its structure. On the left, you have this DNA binding domain, and that's the domain that actually goes to bind to DNA in the same place where the duck was on that previous site, on that previous slide. And then on the other end of that protein, you actually have this transcriptional activator domain that signals to the whole machinery of the cell to start reading those downstream genes. So just wanted to, now we'll launch into a brief overview of our project, but just wanted you to have that biological context to guide that understanding.
There are over 20 drugs that are currently in the FSHD pipeline. Unfortunately, there is no approved therapeutic that's on the market for public use, but there is definitely hope. Here are the key players that you can see on the right. When it comes to FSHD research, there's rapid progress, and every day there's something new that's coming out.
But yeah, so with our project, we hope to develop some a form of a treatment that would be personalized and accessible through the mechanism that Heloise was describing. And this way we would reverse the progression of FSHD. And so there are two isoforms, as Heloise was saying, the long form and then the non-pathogenic short form, or the DUX4 DVD. Does someone want to take these slides?
Sure, I can talk a little bit more about this. So the structure that was shown in the earlier slide is actually the structure of the full transcription factor, the full DUX4 protein. And that's what's expressed in FSHD patients, is this protein that has a site that, part of it that both binds to and grabs DNA at specific sites in the genome, and another part of the protein that kind of grabs ingredients from the cell to read those recipes and create those toxic cookies. And so our approach actually centers around overexpressing just a short form of that protein, just the DNA binding domain that will sit at all those sites that the full-length DUX4 would sit at, but not have the capability to actually recruit the ingredients to the recipe. If you want to keep going, Alice.
Yeah, so we plan to basically assess the performance of this short form without that harmful long form, just to cut it out and basically see and assess this performance in cells by a co-transfection. So we're going to start our testing in HEX cells, but we want to ultimately apply this to muscle cells and then later into patients. But we hope to just kind of test the correct expression ratios that we would need. So we would experiment with what's the proper amount of the DNA binding domain form that we would need to add.
Yeah, so another part of like our project is basically inducing the fusion of respective like DUX4 long form and DUX4 DNA binding domain, which would be expressed in C2, C12 mature muscle cells, which are, they basically form the basic structure of like elemental, like skeletal muscle. So with the C2, C12 myoblast or like cells, I guess, they would fuse together into myotubes upon differentiation. And so essentially we would just want to express both of them together after expressing them separately.
And a final very exciting avenue of the project is computational modeling. And our team noticed that like a big challenge for FSHD is that there's a lot of clinical variability. So we were wondering, how can we make this treatment or this solution very fine-tuned to the person, or how can we know or can predict the effects on a human body in real time? Next slide. So what we were planning to do for computational modeling is doing a prediction model, and we're basically planning to use diverse databases from the different types of genomic activity in different patients to then possibly fine-tune that amount of overexpression in specific patients, and hopefully take that next step toward that personalized treatment that we're looking for. So that's also very exciting having it to consult.
So that's just a brief overview of our project, and I want to thank you all for listening to us present about this. We're very excited because essentially the idea behind our therapy is that this is the only therapy that we know of that's kind of in the works that would both address stopping DUX4 in its tracks, so stopping progression, but also enabling muscle regrowth through that kind of fusion experiment of muscle cells. And so this is a really exciting avenue, and we just started this out, and so there's a long road ahead, but I know I feel very motivated to keep working on this and to keep advocating and researching until a cure flows through the veins of every patient. And I feel very lucky to be working alongside a team of such brilliant, compassionate, talented, and motivated individuals who have such amazing and diverse skill sets and such big hearts to dedicate their time and their whole summers to working on this project. So we have an Instagram account if you want to follow along with us, if you have social media over the summer and as we continue working on this project. But yeah, that's kind of our spiel, and we're open to taking any questions now.
Heloise, I have a question for you. So, and again, this might sound dumb because I don't know anything about all this stuff necessarily. When you're doing these experiments, do you have a muscle biopsy from someone that has FSHD? Is that what you're experimenting with?
So in the lab, the scope of our project definitely relies on cell work, because that's kind of the first step of exploring a new drug, is first starting in cells and then potentially moving to mice and then potentially moving to other animal models and humans. And there are many different cell lines out there. We are starting off with a cell line called HEC cells, which is basically just a very general human kidney cell line. So it's not very specialized around muscle, but it's such a great model for validating reporters and other components of our research that will be later on applying to other cell lines. And so basically the next model that we would move into that we have planned is a model called C2C12s, which we briefly mentioned as well, which is actually a muscle cell line isolated from mice. And so that forms a great model for us to better understand how cells will fuse together and how muscle behaves in a more reliable way. And then next we would really hope to get access to a cell line isolated from FSHD biopsies, because obviously that would be kind of the most reliable model for DUX4 expression, especially because DUX4 is so kind of spontaneous. But there's a lot of paperwork that goes on behind getting a cell line like that. So we're getting our experiments started in these preliminary models and then hoping to move into those more specialized cells later.
And how long will this process take?
That's a really hard question to answer. Science can move really quickly or it can move really slowly. It just depends on if your experiments work, if your cell lines cooperate. Right now, for the next couple of weeks, we're all full-time students at Stanford. So we have actually started in the lab last weekend, but things are moving a little bit slower than they would because we're all, loaded with classes and tests. But we'll actually all be working in the lab full-time this summer together. So things will be able to move much more quickly then because, well, we have 8 team members working full-time and we're hoping to onboard a couple of interns as well. So it's hard to tell how fast it'll move, but we're doing all that we can.
I know you're just a researcher now, but your ultimate goal obviously is to, and you stated this, is to get this this treatment into as many patients as possible. As you well know, you're an early onset, pediatric onset FSHD, and we've been advocating to get it to the younger patients, while they still have muscle before their muscle deteriorates. So can you tell us how you would feel, how do you envision your research going into clinical trial and what kind of population, that you would target for that? And what's the importance of getting those, how important is it to get the muscle when they're still pretty healthy before DUX4 has had a chance to really deteriorate them, like in the younger patients before the disease had a chance, had taken its toll, so to speak?
Yeah, those are all really great questions. So right now we are in the very, very early stages. I mean, we just started in the lab last weekend. So it's hard to know exactly how this will pan out through clinical trials because that would be likely years down the line. But right now we're exploring this idea with the hopes that 1, it'll work, and 2, that if we will definitely kind of move forward with this idea in one way or another. So one thing that we're exploring right now is actually a big grant that would provide a lot of infrastructure and support for us to go into a potential startup with this idea. This is just kind of floating in the air right now before we really gather more data. But this is the very, very start of drug development, and there are many years that would have to go by before it actually gets into patients. And so since we're just starting out, it's hard to tell exactly how that'll pan out, but you know me, I'm very dedicated to getting a treatment on shelves as soon as possible. And so I'm absolutely on your side always for advocacy, to make sure the kids' voices are heard and that they're not forgotten and left behind in the process. And so, in the meantime, the exciting thing is that there will probably be a drug on the market before this one even comes into clinical trials. But the challenge is going to be, to advocate and make sure that that drug is available to everyone before ours actually hits shelves.
Heloise, that was a really great presentation. It's nice to see you, and congratulations with your team. And it's nice to see your team that you're working with on this.
Hi.
For
Didn't
those of
see you
people who
in
don't
the-
know me, yeah, nice to see you. I'm a physical therapist, but my real job really is in clinical trials, natural history, and drug development, particularly looking at outcomes for neuromuscular diseases, and in particular FSH as one of our primary areas of interest here at the neuromuscular division. So one of the things is, first, Ali asked you a really hard question. So great job answering it. I just had a few, one of the questions you semi-answered. One was like, in development of these cells, are you looking at iPS cells in which you need human biopsies? Just because one of the things that may be useful is to see where our paths parallel or align, that we could help you with any samples that you may need, and how we could further that along with some of our natural history studies that we're doing with FSH, in which we are taking muscle biopsies, or if there's surgery that we may be able to get samples from surgery, and we have a biobank for that. So just kind of put that in the back burner as you guys are moving through things. My second question is about the genomic databases. When you say fine tune, what databases are you guys looking at? And I know I missed the very beginning, so you may have already said it. When you're looking at fine tuning, are you looking at some AI and machine learning strategies, and targeting that specifically?
Yes, I think I can hop onto that. Thank you for your question. About the genomic databases, we've read past literature about it, and there's some modeling work that has been done for the past couple of years, and they use very robust like genomic databases. So we're planning to get onto those and kind of updating other ones, because most of these were made in 2021, 2022. So we wanted to update these and kind of also combining the results from the lab itself, because that's also going to be like new data to make it more of a predictive model, right? But yeah, thank you I don't know if that answers your question. But
yeah, no, but I guess my other question alongside that is that, the personalized medicine approach to it. So would the genomics be predictive of the ability of a muscle to regenerate? Because it sounds like the second half of what you're trying to do is to target the downstream effects of FSHD. So you need to have some muscle fibers that are intact if they're in the inflammatory phase or not. I'm assuming or guessing. Is that what you guys are?
yes. So the idea for computational modeling is to aid wet lab to find that ratio, because patients have different like copies of like DUX4, right? So like if we find that amount of overexpression, we want it to be as specific as possible depending on the genomic data that we have. So I feel that the general idea of that is to kind of make a type of prediction, like how much do we need to overexpress to counteract the aberrant expression of DUX4 full length. That's the main goal.
I wanted to say thank you so much, Tina, for the offer about the biobank, because we'll definitely, as we kind of move deeper into the project, we'll definitely have a lot of use for more reliable cell models of FSHD. And so that's definitely something that we'll keep in mind
Yeah.
in the future.
No, for sure, because what you're describing is we actually work with a few basic science labs around campus in doing very, similar things, looking at gene therapies and different things. So we work kind of in parallel, kind of a translational medicine approach, and to ensure that if you need samples, that we're able to get it, and when we have patients, we can include that as part of, you come see us how it works. We have a research team that always says, hey, we have this and this project if you want to be a part of it. So if we can help in that way, and anticipate what you may need, we can start collecting that now.
Wonderful, thank you.
We have a question from Shivani. You want to ask the question?
Hi, yeah, hi. I put a question. I see that Emmanuel also just sent me a direct message. I don't know if maybe it would be great to just let him speak more on it, but I just saw the direct message sent to me. I guess I'll just say my question anyways, and then let Emmanuel take it away. And then I also actually had a second question. Also, I just wanted to say, like, this is such a great and exciting project, and I really commend all of your team for taking initiative on this, especially at such an early stage. So thanks for your dedication to the cause. And then my other question, apart from the one in the chat, well, my one in the chat is, can you elaborate on how the fusion of skeletal muscle cells may lead to regeneration of fibrotic muscle? For example, if a sick or fibrosed muscle cell fuses with a healthy one that is not expressing as much DUX4, like, what do you think the outcome of that would be? Because I know you correctly pointed out that there's no current therapy that really targets regeneration. So that's a really exciting angle of your project. And I also realized that the difficulty kind of with fibrotic muscle is that it's really hard to reverse the fibrosis. So I'm just curious what your thoughts are on that. And then I have a second question. I don't know if you want me to wait and ask that one or just ask it now. I guess I can just ask it now. My second question is about, so I'm sure you guys are very well aware already that DUX4 is only transiently expressed. Like, it's pretty sneaky in that it's like not constantly expressed. It's only expressed in bursts, in like very sporadically. So that makes it one of the hard things to target. And looking at the outcome of the current clinical trial for losmapimod, one of their primary outcome measures in like their early phase clinical trials was looking at a reduction in DUX4 expression, which they actually did not find. So I guess it might just be, and I'm sure you guys have already considered this as a potential technical challenge, but I'm guessing what is kind of your thought process on navigating that if it comes up as a barrier, and what other outcomes or measures do you think we could look at other than DUX4 expression in case that becomes an issue like it is in the losmapimod trial?
Well, I can answer the first question. So we plan to include like a muscle promoting factor in our therapeutic. This could be like a myostatin inhibitor, or maybe something else that works better in this situation. And we aren't exactly sure if we expect to see growth from the fusion of a myostatin inhibited muscle group and FSHD affected muscle group. But we would like to test for it to see if it does lead to muscle growth. And if it does, we'll include it in our general project.
Yeah, and to kind of add on to that, our kind of big picture hopeful approach would incorporate this kind of like muscle stem cell technology incorporated with our own therapeutic. And so one experiment that we're really gearing up for and excited to look into further is kind of this fusion of C2C12s just for now, just because they're kind of easier to work with. But C2's expressing full-length pathogenic DUX4 and C2's expressing our DNA binding domain, and seeing, including a reporter that we were able to get from another lab, and seeing what happens when we fuse these samples together. Like, for example, in the cells that are endogenous, that we induce expression of full-length DUX4 in, once they fuse with our DNA binding domain isoform, does the downstream expression of those downstream genes actually go down in those very muscles due to the multinucleated nature of muscle fibers. I'm having trouble explaining this without a graphic in front of me, but does that kind of make sense? It would essentially be harnessing the potential for muscle stem cell therapy, which I know is very far out in the future, just in the general space of muscle research. But because we have a long road ahead of us, and that would kind of be the ultimate dream. And as Emmanuel mentioned, we're also looking into potentially including myostatin inhibition in our assays as a potential for muscle regrowth. And then to address your second question about the sporadic nature of DUX4, that's definitely something that we're keeping in mind, especially because a lot of our early models are more so focused on validating whether the binding domain can actually act as a competitive inhibitor of DUX4. And so as we move into more experiments to better model a reliable FSHD, to better model what happens in FSHD, we will be keeping that consideration in mind. But in general, I do want to be mindful of my teammates' time, because I know that they have exams. So guys, feel free to hop out if you need to get back to studying or schoolwork. And I know that there are a lot of parents of early onset patients who would love to be part of the discussion as well. So would love to open up the floor to any more discussion.
I have a quick question. So my daughter has type 2. Would that, is there any research between the type 1 and type 2, or is it all together? I guess I'm not sure how that would work.
Yeah, great question. And that's something that we were thinking a lot about while we were brainstorming ideas, because we wanted to include as big of a population of patients as possible. And so the exciting thing about this therapy is that hypothetically speaking, we hope that this would actually address both FSHD1 and FSHD2, because the difference between FSHD1 and FSHD2 genetically is just the type of mutation in the DNA, and both of them lead to the expression of DUX4 that should be silenced. And because our drug or our approach is actually centered on competing directly with DUX4 once it's expressed, it should address both FSHD1 and FSHD2. There is a little caveat in there, in that DUX4 is very sneaky and actually goes on to modulate its own expression. And so that's where we're not sure exactly how the pathway would play out, but we're definitely keeping both FSHD1 and FSHD2 in mind as we move into our experiments.
Thank you.
So you mentioned that it takes a while for any treatment or drug to go from the research to being, I guess, commercially available. Typically, what timeline are we looking into from where you started? I guess you said for this last week. Do you guys have like an average timeline?
It's that's an excellent question, and it's really hard to say at this point, because we're all freshmen and sophomores at Stanford. So we still have a lot of an educational journey in front of us. We're full-time students, and so because of that, things move a little bit slower than if we were all working full-time in the lab. And so again, like, we are in the very beginning stages, these drugs that are in the pipeline right now in phase 2 trials, they have been in the works for years. It's hard to say exactly how many, but probably, I mean, June might be able to answer this better than I can, but maybe like 10 or so years of people working full time and dedicating their full careers to this. So, we're a team of students with a passion and a desire to make a difference in this field. And so we're doing all that we can to get a treatment to shelves as quickly as possible. And I will never stop working closely with the patient community to advocate for that treatment to reach shelves for everyone as quickly as possible. But as of now, it's hard to tell, if this research works, which we have to see if it works, first of all, to know like when it might be available.
Are there any other questions for Heloise or her team?
Will you guys be attending the Connect conference this year?
We actually have our finals like right around the same time as the conference. So unfortunately that makes it really, because we're on a quarter system here. So our school just runs into mid-August. So unfortunately won't be able to make it to the conference this year. But we will be participating in World FSHD Day from afar. We were actually just talking about that yesterday and earlier today. So we will be there in spirit, absolutely.
I have two questions. One is just out of curiosity. Was there a department there at Stanford that had already started this research, or was this you taking the lead? And also thank you so much, because you are inspiring and it's amazing everything that you're doing. My second question is about your experience just to get around campus and you conserving energy and how you do it, and how accessible is it, and how does a university provide supports and resources?
Absolutely. So to answer your first question, there was actually no faculty member working directly on this project, but this was a research idea that I've had kind of floating around for a while and have wanted to try out. And so there is a great support system for our iGEM team through Stanford. We have lots of wonderful mentors, and there's an amazing lab that's specialized in muscle that I actually have worked with previously, that is able to provide us with cells and a lot of knowledge as well. But so the specific idea and FSHD research itself was kind of novel here, and I'm really excited to be working with this team of really talented and amazing individuals alongside me. And I feel really humbled by their support, not only in the lab, but also just personally, and you guys are awesome. But to answer your second question, so the Stanford campus is pretty big and pretty sprawled out, and that is something that really scared me when I came here, when I was deciding which college to go to. But there's the advantage that it's flat, which definitely makes it much easier to get around. And so when I was making my college decisions, another thing that I was really taking into account was that it's California, which tends to have a lot more, there's a lot of advocacy for, like a lot of disability advocacy and support here. So that definitely made me feel more comfortable. And I basically at Stanford, like 99% of students get around with a bike. I'm not able to bike anymore, but I invested in an electric bike, which helps me get around the campus much more easily. And there are elevators in like all the buildings that I need to access. And there's an office called the Office for Accessible Education that provides many resources, both in terms of housing and in terms of academic accommodations, to help support me in the ways that I need support. And I will say there's always more work to be done. I had an incident earlier this year where I felt really frustrated with administration. I won't go into the details, but it was surrounding clinical trials and my participation in clinical trials, and basically my professors, this Office of Accessible Education, made it really hard for me to be able to sign up for a clinical trial and like compromise some of my academic commitments. And so in the meantime of trying to figure that whole thing out, the spot in the trial was actually given to someone else, and that was extremely frustrating. It was a couple months ago now, but it still brings me to tears every time I think about it. So that just goes to show there's a lot more work to be done. But yeah, we are getting angry.
Thank you for answering. Just one more thing. Does your how do you get through the workload, or is that modified for you, or do you just plow through it?
That's another great question, and one that I should ask myself more often. I am definitely the type of person who tends to overload my plate, just because there's so much I want to do, especially with FSHD and now this really exciting research project. I want to do all that I can. Every day matters. Every minute matters. And so I have to remind myself to slow down sometimes, because on top of that, I'm taking a pretty charged academic load with bioengineering and pre-medical studies. But I am trying to learn day by day to give myself time and breaks. Luckily, all of my professors are very understanding. I have personal conversations with them at the start of each quarter, which really helps explain my situation, and the fact that there's no cure, so sleep is the closest you get to a cure now. And so yeah, I try to work with my professors, and I'm lucky that most of them have been understanding. But it's important to take breaks, because I have a hard time reminding myself that my health is the one thing that sticks with me the rest of my life, and with FSHD, there's no going back. So.
Heloise, I met you several years ago, and you were leading the Florida chapter and doing great things, and you were still in high school. So ever since I've known you, you've been advocating for FSHD. Can you speak a little bit to how has that helped empower you, and how has that affected your mental attitude and your drive as a young person with this disease? Can you speak a little bit to that, how advocacy how has that impacted you, and how has that helped you deal with having FSHD?
Absolutely. That's kind of that's made all the difference. When I was first diagnosed at 13, that really flipped my life around. I was a spontaneous mutation, so no family history, had never heard, scapulo-humeral disease, like what does that mean? As I'm sure many of you have experienced in your families. And this just came out of nowhere. I was a seemingly healthy child up until my symptoms started showing up, and my sports were everything. I was super athletic, riding horses, playing tennis all the time, and then all of a sudden this ball dropped. And I was attending a really athletic school district in Texas, had just recently moved from Belgium. So there were a lot of changes happening in life. And then this just kind of put a stop to everything. And the first couple of years after getting diagnosed were indescribable. Like I could never go back to that time. I suffered immensely with my mental health. Suffered with extreme anxiety and depression after my diagnosis, and it seemed like nothing that anyone could do or say or try to put into my body made a difference, until one day I sat down, and I remember this day so vividly. I was in my living room, and I was like, you know what? I've heard about this FSHD Society, and actually, before that, I forgot to mention, June and other members of the FSHD Society had actually come to my house and to meet me and introduce me to the wonderful community of advocates and researchers that are working every day to make a difference. And so one day I sat down and I was like, you know what, I've heard about this FSHD Society thing. Let me just do a little bit of research, whatever. And I watched this webinar about ways to get involved in the FSHD community, and I felt this gripping feeling inside my heart. It was like, this is something I have to do. Like, why? I can't just sit around and wait and wallow in self-pity while other people are out there raising their voices and standing up for those with our disease who literally can't stand. I can't just sit around. I have to get involved. And so that was the day when everything changed. Like that very night, I sent an email to Beth Johnston at the society and said, I'm in, I'm raising my hand. I want to start a chapter here. And at the time I was 15 and my parents were like, whoa, like, do you want to, like, you've been really struggling with this for 2-3 years. Do you want to like take a night, think about it, sleep on it? Like, are you sure? This seems like a big commitment. And I was like, no, like I have to do this. You can't stop me. And so from that day, I like to say that I've become kind of addicted to activism. And it's truly made all the difference. And I feel like by becoming an advocate in the community, I'm not only advocating for myself, that's just such a small part of it. It's really about advocating for something bigger than myself and for a community of nearly 1 million people worldwide who are living with this disease every day. Again, standing up for people who physically can't stand, making sure that no child in the future someday will have to fear losing their independence, because that's something that I wake up every day thinking about, like when will I not be able to live independently? And when I was going through my diagnosis process, I was told by a pediatric neurologist that my pain was due to my teenage imagination. So that too, making sure no other child must hear that their pain is made up. This is about joining a global movement that is, changing lives and eradicating this cruel disease. And so that's really what starting research has meant for me as well, is expanding to not only advocating for myself and others, but also kind of holding a stake in the day, in accelerating the day that a cure comes.
Thank you, Heloise. That was very well said. And I'm glad you said it in this forum when there's, a lot of parents here and we all have busy lives and we're taking care of children that need, a lot of care. And I'm glad you said that, because me as a parent too, it helps me, like I'm doing something, and I think a lot of parents feel that way. And I think our kids can get involved, and it helps them to know that they're doing something to help themselves and the others that will follow them. So you've always been a shining example to the whole community, and we thank you so much for everything you've done in the past and what you're doing now and what you will do in the future. We thank your team for being here, and thank you so much for taking on this, this tremendous task. And this is a very hard nut to crack, as you all well know. And we appreciate your efforts so very much in doing that. And you guys are all young and brilliant, and I know you'll do great things, and you could have done anything. And we appreciate that you picked FSHD, to put your time and effort and brilliant minds to. So thank you all so very much. On behalf of all the FSHD community, the parents, the kids, and all the adults that unfortunately suffer with this disease. So thank you all so very much. And we plan on having you back either in this forum or in another forum so that you can give us your results, give us some data and what you guys have found out. And we're looking very much forward to hearing what great things you guys are doing. Kristen, do you have something to say?
No, just extremely impressed always, Heloise. Great work with you and your team. And thank you for always continuing to fight and get angry.
Get angry.
and angry.
A wise little bird told me that one day.
In all the right ways. Put the anger in the right channels. So
Productive
that's
anger.
exactly what you're doing.
June, would you like to add something before we wrap this up? And I know these.
Oh, I just hope to see a poster at next year's International Research Congress from your team. That would be cool. And of course, you know, many of us are here to help support you in this work. If you want to connect with any researchers out there or any, you know, advice on, you know, as you move forward with this research, we're all here for you.
Yeah, and I hope you make a company out of this for the future FSHers that are coming along and the generation behind you as your little future workers.
FSHD STEM for kids.
Yep.
Well, I wanted to say also that, you know, I know this is the early onset parents roundtable, and I'm sure that a lot of you in the call today are either early onset FSHers yourself or parents. And to the parents, I want to say that similar to what Ali just touched on, I'm sure that some days it can feel frustrating and like a cure is so far away and you want to do everything you can for your kid, but every single day that you advocate and every single day that you support your child and put your voice out there and get angry and advocate to the government and just share your FSHD story, that really makes a difference. Sometimes it feels, when I work on my advocacy projects, it feels like just a little drop in the ocean or a little drop in the bucket. But I know it sounds cliche, but every one of those drops is contributing to a whole ocean. And so nothing that you do goes unnoticed. And every single day that you're advocating and supporting the community is making a world of difference. So thank you for all that you do for the community and for just being here, staying up to date on the research, you know, even just listening to a group of college kids present about their research, you know, thank you for being here.
You're quite welcome, and we look forward to hearing more about this project in the future. That's our hour. I want to respect time. I just want to mention real quick before we wrap up, the Connect conference is coming up in June. We have a lot of activities for teens and kids at Connect, so there'll be more to follow on that. We're also going to have an early onset breakout, and we would really like the families to come there. We're going to have a panel of experts. And also there's going to be a lot of pharma in the room. So it's a great opportunity for you to ask questions. I know a lot of you may not be close to a CTRN and you don't get to see a FSHD expert every time with your child. This is a great opportunity to ask an expert, to ask the pharma, to advocate for your child and for trials and treatments for those that are being left out right now, the kids and the non-ambulatory. So we hope to see you all there. If you have questions, if you can't make it to Denver, but there's a burning question you want to ask to a researcher or a pharmaceutical on behalf of yourself or your child, please shoot that to me and I'll make sure that I ask that question on your behalf. I'm at early onset chapter at fshtsdsociety.org. Please shoot me an email and I'll make sure to ask that question for you. Marissa, do you have a quick question before we go?
Yeah, since you're talking about the conference, I just wanted to plug that we're also doing the conference is also virtual.
Yes.
So if you can't make it to Denver, like I can't, we're doing a virtual thing as well. So you'll be able to watch all of the big panel discussions of the conference. And then we're doing our own little breakout room sessions, a little bit of a more introspective take to the breakouts. So I just wanted to plug that real quick. Thank you.
Great reminder. Thank you so much.
Thanks,
Thank you so
Marissa.
much. Yeah. So yeah, if you have any questions, again, if you want to find out more information, I think registration is closed now, but
Well, the in-person, but the virtual Connect is
Okay.
still open. So
Okay, virtuals to open. And there's more to come. We just did a trip to Washington, DC, and the FSHD Society has published some stuff on that. We did have a lot of early onset representation. I think we had 3 early onset families. We made sure that the representatives and the people we spoke with knew that this disease affects people of all walks of life and all ages, and the kids and the early onset definitely need to be represented. They definitely need a pathway to treatment. So there's probably going to be more to come on that. I think there's a couple articles that are coming out. I know the FSHD Society is publishing some more stuff. So I just want to let you know that we are out there at every chance we can get, yelling for the kids and the non-ambulatory and the early onset, and that we will make sure they are not forgotten.
Yeah.
So I will let you go. I know. Go ahead, June. You want to?
And this every year, in September, we have a walk and roll to cure FSHD, and there are walk and rolls all over the country. There's certainly one in the Bay Area chapter, and that's another way. As always said, it might seem like a drop in the bucket, but collectively, when you know that you're out there walking, maybe in a small group, but across the country, there are thousands doing it. It's amazing.
And this is amazing community, and just getting to meet other people, because you may go, most of your life and never meet another person with FSH, but these sorts of events, give you an opportunity to meet other people and form bonds and friendships. So if you can support it, please do so. All right. So for real, I'm going to let you guys go. I don't want to call you kids. I know you young folks got to get after your studies. So thank you so much for
Good luck
being here.
yeah.
And thanks everybody for attending the meeting tonight. I greatly appreciate it. And everybody have a great Memorial Day weekend.